Okay, let's unpack this. We are about to get into a really incredible story of, I think, scientific persistence. I
hmm
mean, if you were looking for a biography that combines just foundational genius, this dramatic professional isolation, and then this, ultimate massive vindication, you found it.
Yeah, this is the one.
This is definitely the one. We're doing a deep dive into the life and work of Barbara McClintock, an American scientist and cytogeneticist whose journey really fundamentally changed how we view life itself.
And McClintock, who lived from 1902 to 1992, is just one of the most remarkable figures in 20th century science. Period.
No question.
So our mission today is really to trace her career, which led to a discovery that completely overturned a central dogma of genetics.
And when you say dogma, you mean something that was just taken as, like, bedrock fact?
Absolutely. The deeply held belief that the genetic code was a static, unchangeable blueprint. She just shattered that.
Right.
She proved that the genome is, in fact, this dynamic, fluid, and often self-editing system.
And the crowning achievement for this came so late in her career in 1983 when she was awarded the Nobel Prize in Physiology or Medicine.
Right, for this stunning breakthrough.
But here's the detail, and this is the one that really underscores her singular genius for me. As of today, 2025, she is still the only woman to have received that specific Nobel Prize entirely unshared.
It was hers alone.
Hers alone, for discovering what she called mobile genetic elements.
And that core concept, mobile genetic elements, is the one that took the scientific world decades to really accept. We're talking about transposons.
Or, as they're more dramatically known, jumping genes.
Exactly,
Yeah.
jumping genes.
Mhm.
Before McClintock, the consensus was that genes were just locked into these rigid positions along a chromosome.
Like beads on a string.
Perfect analogy. Passed down in this orderly, predictable fashion. Her discovery showed that segments of DNA, these things she called controlling elements, could literally cut themselves out and move to entirely different locations.
Wow.
That movement is called transposition.
That intellectual fight, I mean, the conviction you would need to stand by an observation that just contradicts the basic physics of your field, it requires a very unique personality.
Oh, for sure.
And you see glimpses of that right from her childhood. She was actually born Eleanor McClintock.
I didn't know that.
Yeah, but her parents quickly renamed her Barbara because they believed the name Eleanor was too, quote, feminine for her strong, independent, almost fiercely determined nature.
So they saw it in her right from the start.
They saw it, and it really foreshadows a trait that she herself later identified as being essential for her success. She called it her capacity to be alone.
And she needed it, because her pursuit of science was just an uphill battle from the get-go. She gets into Cornell in 1919, but it was despite her mother's really significant fear.
A very real social concern at the time.
A huge concern.
Yeah.
That higher education, especially in science, would make her, quote, unmarriageable.
And she chose the science anyway.
She chose the intellectual pursuit over the social expectation, and that pattern just defined her entire career.
So she gets to Cornell, and it turns out to be more than just a school. It's really the perfect incubator for a scientific revolution.
It really was. She earned her BSc in 1923, then her PhD in 1927, focusing on botany and genetics.
Yeah.
But her career pivot wasn't initially, part of some grand plan.
Okay.
We can actually pinpoint the exact moment her future was cast, as she put it. In 1922, a prominent plant breeder named C.B. Hutchison was so impressed by her talent in his genetics course that he made a simple telephone call. Just invited her to participate in the graduate genetics course. It was a huge opportunity.
And she always credited that single call.
Always. She said, it cast the die for my future. That one call was the catalyst that defined her entire professional life.
And that one act sets the stage for what we now recognize as the golden age of corn genetics at Cornell. She immediately became the central figure in this incredibly influential group.
A group focused on this emerging field of maize cytogenetics.
Right, and the circle she was in included future giants like Marcus Rhodes, Harriet Creighton, and even George Beadle, who would go on to win a Nobel Prize himself.
And if we just pause on that term, cytogenetics, for a second.
Please.
It's the perfect description of her expertise. It's a meeting point of cytology, the study of cells and their physical structure, and genetics, the study of heredity.
So she was the person who could physically see what the gene was doing under the microscope?
Exactly. She could see it. And what's so fascinating about her work in this period is that she didn't just study maize. She literally invented the tools necessary to study it effectively.
She was a methodological genius. She developed these crucial visualization techniques that made these tiny, tangled Maize chromosomes legible for the very first time.
She used carmine staining, for instance, which was a method that made the chromosomes appear much more clearly defined. But the real ingenuity—
It was where she looked, right?
that's absolutely key. The standard practice at the time was to study chromosomes from cells taken from root tips. McClintock shifted her focus entirely to the cells from the microspore,
Male reproductive cells.
the male reproductive cells, which are
Okay.
undergoing meiosis.
So why was that the game changer? Why the microspore?
Because of the clarity. During meiosis, the chromosomes pair up and they exchange material. So by studying the microspore, she could visualize the chromosomes at a much cleaner, less cluttered stage of cell division.
So she could actually see what was going on.
She could see it all. And this allowed her to identify and characterize the specific morphology, the size, shape, and structure of all 10 Maize chromosomes.
All 10 of them.
All 10. This work alone became the absolute foundation for all subsequent Maize research. It ended up in genetic textbooks for generations. She literally built the map.
And once she had this map and these incredible tools to visualize what was happening, she could immediately start to prove or disprove genetic fundamentals that had only existed as theories before.
And the breakthrough that really cemented her and Cornell's reputation came in 1931.
The paper with Harriet Creighton?
Yes, the publication she co-authored with Harriet Creighton. This was the seminal work that provided the first indisputable physical visual evidence
Visual evidence.
linking chromosomal crossover, the physical exchange of segments between chromosomes during meiosis, to the actual observable recombination of genetic traits that Mendel had predicted.
Okay, so before this paper, the idea of crossing over was just, what, a necessary hypothesis?
It was a mathematical necessity. It was the only way to explain why offspring had combinations of traits that weren't present in their parents. But they saw it.
They saw it happen.
They correlated a physically visible exchange of chromosomal segments with the predictable shift in inherited characteristics. And that moment changed the concept from a compelling theory to a proven, visible mechanism of inheritance.
Wow.
And beyond proving recombination, she dedicated this period to characterizing the fundamental physical architecture of the chromosome itself. These are the structures that are essential to maintain genetic integrity.
Right, so like the centromere.
For example, the centromere. In 1938, she published this incredible cytogenetic analysis of it, detailing its organization and its dynamic function during cell division.
The centromere being the structure where the spindle fibers attach to pull the chromosomes apart.
Exactly, ensuring they're distributed evenly. She also successfully pinpointed the nucleolus organizer region, or NOR,
Okay, and she located that where?
precisely on Maize chromosome 6. And this is the genetic region required for the assembly of the nucleolus, which is the central hub for ribosome production in the cell.
But maybe the most stunning thing from this early period, she began deducing the existence of structures she couldn't perfectly see, but she knew had to be there based on what she was observing.
Right, which led to her early hypothesis about the structures that protect the tips of chromosomes, what we now call telomeres.
This is such a classic example of her foresight. How did she figure that out?
By observing what happened when chromosomes broke. When a chromosome breaks, the broken ends are highly reactive. They fuse with other broken ends. So she reasoned that if the ends of a natural, healthy chromosome were the same as those broken ends, the genome would be inherently unstable.
Chromosomes would just be sticking together end to end all the time.
All the time. A total mess. So she figured the natural tips must have a special capping or protective function to prevent fusion and ensure stability.
And this was decades before we understood the molecular chemistry of telomeres.
Decades. She was deducing a molecular structure purely from observed cellular behavior. It's remarkable.
And despite achieving this high watermark of technical and theoretical science, the institutional hurdles for women in academia at the time just became immediately apparent.
Yeah, she secured prestigious postdoctoral fellowships, including a Guggenheim, which took her overseas for a bit.
That Guggenheim led her to Germany, right?
Right.
She was planning to work with Kurt Stern.
She was. But Stern, who had also confirmed crossing over in fruit flies shortly after her own paper, had already fled to the U.S. because of the escalating Nazi political tensions.
So who did she work with?
She ended up working briefly with Richard B. Goldschmidt in Berlin, but the environment was just too volatile, and she returned early for the same political reasons.
And when she tried to settle back at Cornell, the very institution where she had essentially invented the field of Maize cytogenetics.
They wouldn't offer her a permanent faculty position, not one that was commensurate with her genius.
Primarily because she was a woman.
Primarily because of her sex. She was supported by grants, worked as an assistant, which eventually led her to accept an assistant professorship at the University of Missouri in 1936.
A transition that marked a move, but certainly not an end to her institutional struggle.
No, not at all. Her move to the University of Missouri in 36, though, it did open the door to her next major set of discoveries.
Right, this is where she meets Louis Stadler.
Louis Stadler, an important geneticist there, who introduced her to a tool that would completely change her research, using X-rays as a mutagen.
And using X-rays to induce genetic changes that was revolutionary. It allowed researchers to artificially crank up the rate of mutation.
Far above natural levels.
Yeah.
It gave them a steady stream of variation to study. It was like taking the natural process and, cranking the dial up to 10.
So by cranking that dial, she starts seeing these incredibly destructive, rapid-fire genomic changes in the irradiated maize cells.
Right. This phenomenon, which she systematically documented, is the breakage-fusion-bridge cycle, the BFB cycle.
Okay, let's unpack this mechanism step by step because it really shows both the extreme fragility and the extreme resilience of the chromosome.
It really does. So it all begins with chromosome breakage. The X-rays would just shatter the chromosomes into fragments. She focused on the fragments that still have the centromere.
Right.
When these fragments replicate, they become two chromatids. But the raw, broken ends of these chromatids are super reactive.
So they want to stick to something.
Exactly. And during the next interphase, the broken ends of the sister chromatids, they fuse together.
So now you have these two chromatids that should be separate, but they're literally stuck together at the raw ends where the protective telomeres used to be.
Precisely. So when the cell enters anaphase, the stage where the chromatids pull apart toward opposite poles, this fused structure forms a physical chromatid bridge.
A bridge across the dividing cell.
A bridge. And as the spindle fibers pull harder and harder toward the two poles, the tension becomes too great, and the bridge inevitably breaks again. But, and this is the key, the second break happens randomly, not at the original fusion point.
Which means the two new daughter nuclei both receive genetically unstable, mismatched chromosomes, each with a brand new, reactive, broken end.
And the cycle repeats. Those newly broken ends are now available to rejoin in the next interphase, forming a new bridge in the next mitosis. It just keeps going.
So what's the result of this?
The result is massive, rapid, large-scale mutation deletions, duplications. And she was able to detect all this visually as variegation.
Unstable patterns of color and texture in the maize kernel.
Yes, in the endosperm tissue. It looked like a mosaic.
This sounds absolutely devastating to the cell lineage. But the discovery was profound for two reasons that go beyond just, observing a mess.
It was. First, she demonstrated that chromosome rejoining after breakage is not a random event. The broken ends were actively seeking to rejoin. It was a specific process.
That process we now know involves DNA repair pathways.
Exactly. And second, it proved that this cycle was a powerful, ongoing source of massive, large-scale genetic rearrangement within a single organism.
And what's fascinating here is that the BFB cycle, it isn't some historical curiosity. It's intensely relevant today.
Oh, absolutely. This mechanism is one of the primary drivers of genomic instability in human cells, especially the rapid generation of genetic variation that you see in various advanced cancers.
So she was observing the mechanism of tumor evolution decades before we could even sequence a tumor genome.
Decades before. It's incredible.
But despite the staggering scientific output she was producing at Missouri, this period was just marked by this deep professional dissatisfaction.
Yeah, she felt completely excluded and marginalized. The sources say she was often left out of faculty meetings, and she perceived this very clear professional ceiling.
The glass ceiling.
A glass ceiling. And this feeling of hitting a limit is documented in a very revealing letter she wrote in 1940 while she was actively looking for a new job.
What did she say?
She stated that as an assistant professor earning $3,000, she felt sure that that is the limit for me. The concern wasn't purely financial. It was about stability and recognition.
It sounds like it was a crisis of trust.
It was. She completely lost faith in the university administration. She strongly believed her position was totally contingent on Louis Stadler's continued presence.
So if he left?
If he left, she believed she'd be vulnerable and probably dismissed. And this feeling of precarity, despite her groundbreaking work, is what ultimately pushed her to look for a more supportive environment.
And that search led her away. She took a leave of absence in 1941, and that's when she ended up down on Long Island.
That's how she landed at the Carnegie Institution of Washington's Department of Genetics, located at Cold Spring Harbor Laboratory, or CSHL.
She started with a temporary research position in December of 41.
Yeah, and she was hesitant to commit long-term. She'd been burned by these institutional experiences before. But she found a degree of support and independence at CSHL, and she became a permanent staff member in 1943.
And her scientific merit was recognized almost immediately when she settled there, which just reinforces that her struggles at Missouri were institutional, not scientific.
Without a doubt. In 1944, she was elected to the National Academy of Sciences.
A huge honor.
An enormous honor. It made her only the third woman elected in the Academy's history up to that point.
And the recognition kept piling up. The very next year, she became the first female president of the Genetics Society of America.
And just to prove that her technical virtuosity was still at its absolute peak, she took on this really demanding analysis of the fungus Neurospora crassa.
Right, at the request of her old colleague, George Beadle.
George Beadle.
Yeah.
He was relying on Neurospora to establish his famous one-gene, one-enzyme relationship, but the physical structure of the fungus was poorly understood.
So he needed her to come in and clean up the foundational cytology for his major project.
He did, and she characterized the fungus's karyotype, its complete set of chromosomes, and meticulously detailed its entire life cycle in a matter of months.
Wow.
Beadle, who is usually a very measured man, was just effusive in his praise. He said McClintock did more to clean up the cytology of Neurospora than all other cytological geneticists had done in all previous time on all forms of mold.
That is high praise.
It is, and this feat just cemented her reputation worldwide as the unparalleled master of the microscope, a reputation that really set the stage for the true scientific revolution that was about to follow.
Okay, so now we arrive at the intellectual core of this deep dive. After establishing herself as the preeminent cytogeneticist, McClintock begins these systematic, meticulous studies at Cold Spring Harbor around 1944. And she's focusing on these highly unstable mosaic color patterns she could see in maize kernels. This is the search that led directly to the jumping genes.
Right. The visual phenomenon she was studying, the streaks and spots of color on an otherwise uniform kernel, it suggested a gene was being turned on and off randomly within the developing seed.
So she investigates this deeply.
And this deep investigation led her to identify two interacting genetic loci, which she named dissociation, or Ds, and activator, or AIC.
Initially, Ds was associated with chromosome breakage, which ties back to her BFB work.
It does.
Yeah.
But she quickly realized that Ds had far wider effects on neighboring genes, especially when the other locus, DSAC, was also present in the cell.
And the revelation, the moment that really shattered the static genome concept, came in early 1948.
Yes. She realized that both Ds and AIC could transpose. They were not fixed. They could physically move from one location to another on the chromosome.
Which was an almost heretical concept in post-Mendelian genetics. It requires a fundamental shift in perspective. If you assume the genome is a perfectly indexed book.
She discovered that certain paragraphs could spontaneously lift off the page and paste themselves somewhere else.
Exactly.
And she meticulously described the functional relationship between these two elements. ACK is the crucial master element. It's the autonomous element.
Meaning it can act on its own?
It contains the necessary code to act independently. Specifically, ACK produces the transposase enzyme, the molecular scissors needed for movement.
And Ds?
Ds, on the other hand, is a non-autonomous element. It's defective. It can't move on its own. It needs that transposase enzyme supplied by ACK to mobilize.
So ACK provides the engine that allows Ds to jump. Now let's tie this back to the corn kernel color.
Okay. The phenomenon hinges on Ds acting as a suppressor or an inhibitor. She found that when Ds inserts itself near the gene responsible for synthesizing the color pigment.
In the alerone, the outer layer of the kernel.
Yes, it suppresses that gene, so the seed tissue remains colorless at that site.
But when the activator, ACK, provides the enzyme that causes Ds to move, to jump out of that location.
The alerone color gene is released from suppression, and pigment synthesis can begin. The gene is finally allowed to express itself.
And here is the genius of the mosaic pattern. This transposition event, Ds jumping out, it happens randomly in different cells as the seed develops.
Right. So if the transposition occurs very early in the development of the kernel cells, the resulting cell line that is now producing pigment will be large. It creates a large colored spot or streak.
It's like a cell division clock.
It's exactly like a cell division clock. If the jump occurs late in development, say only a few cell divisions before the kernels mature, the resulting colored tissue will be very small,
A tiny speck of color.
a tiny speck. The randomness of the timing, dictated by the action of ACR and Ds, is what generates the mosaicism she was studying. She even quantified the rate, noting that the number of ACR copies in the cell directly increased the rate at which Ds would jump.
This was just revolutionary. But she didn't stop at color patterns. Between 1948 and 1950, she developed this massive conceptual framework based on this movement.
Her gene regulation hypothesis.
Right. She argued that these mobile controlling elements weren't just genetic debris, they were actively regulating gene action by inhibiting or modulating their expression.
And this conceptual leap was truly monumental. It took her far beyond Maize. She's suggesting that dynamic gene control could be the very mechanism that explains cellular differentiation.
Okay, so think about it. Every cell in your body has the exact same genetic information, the identical blueprint.
Right.
So how does one cell become a neuron and another a liver cell? McClintock proposed that this control was achieved not by changing the DNA sequence, but by dynamically turning genes on or off, temporarily or semi-permanently, via these mobile elements.
And this was decades before the field of molecular biology could even approach such complex regulatory systems.
She reported this groundbreaking work in a 1950 paper, but the reception was devastatingly cool.
She later described the initial response as puzzlement, even hostility.
So why the resistance? Why were they so hostile?
Well, it was a conflict between complexity and simplicity. The prevailing scientific philosophy at the time demanded simplicity and rigidity, the static linear gene model.
And McClintock's system was.
It was complex, dynamic, nonlinear, and it was based on plant genetics, which was often viewed as sort of secondary to the emerging, simpler model systems like bacteria and viruses.
And her observations were just so technically complex, visualizing these changes through cytogenetics. A lot of geneticists steeped in statistical analysis just couldn't comprehend or believe her data.
Exactly. So she had the answer, but the rest of the world wasn't conceptually ready to even hear the question.
So despite publishing detailed statistical data supporting her claims in 1953 and doing lecture tours through the 50s, she just faced profound skepticism.
She did. She felt she risked complete alienation from the scientific mainstream, and this led her to a very difficult, very painful decision. She essentially withdrew.
She stopped publishing.
She was forced to stop publishing accounts of her research on controlling elements after 1953. She chose to work in isolation rather than continuously fight the established paradigm.
So she went silent on her greatest work, but she didn't stop working. She kept refining her model, even identifying a more complex element.
Yes, the suppressor mutator element, or SPM, and the SPM system was even more sophisticated. It demonstrated that the element could fully suppress a mutant gene's expression when it was present, but then activate it again, showing this dual regulatory capacity.
Incredible.
She just continued to document this fluid, responsive genome from her solitary post at Cold Spring Harbor, knowing she had discovered a universal principle, but accepting that the world required time to catch up.
Following that quiet reception of her transposition findings, McClintock found a new avenue for her expertise.
Right. In 1957, she received funding, notably from the National Academy of Sciences, to pivot her field research.
And she started studying indigenous strains of maize throughout Central and South America.
This shift during her so-called silent years on transposition was a massive scholarly undertaking. Throughout the 60s and 70s, she traveled extensively, collecting and analyzing strains, meticulously detailing their chromosomal, morphological, and evolutionary characteristics.
This all culminated in her landmark study, The Chromosomal Constitution of Races of Maize.
A work that not only provided this deep understanding of maize diversity, but also laid the foundational groundwork for ethnobotany and evolutionary biology.
By linking cultural agricultural practices with genetic variation.
Exactly. And while she was focused on this evolutionary diversity in the field, the intellectual climate back home was slowly changing.
Signaling, as she would later say, the right time for conceptual change.
That's the quote. The first crack in the static genome paradigm came conceptually in the 1960s with the work of French geneticists François Jacob and Jacques Monod.
Okay, and they were studying the lac operon in E. coli bacteria.
Right, and they were describing a mechanism where external conditions could turn genes on and off. Their work provided the first clear model of gene regulation, albeit in a simpler system.
And that work conceptually mirrored what McClintock had observed decades earlier in Maize.
Genes being actively regulated, she immediately recognized the parallel. She wrote this influential article for the American Naturalist, drawing comparisons between the bacterial and the plant systems, insisting that dynamic gene action was a universal principle.
But the global validation, the definitive proof that she was right, that didn't happen until the molecular age arrived.
No. New technology finally emerged in the 1970s that allowed researchers to study DNA structure directly.
So molecular biology provided the tools, restriction enzymes, cloning, that were needed to confirm transposition in simpler systems like bacteria, yeast, and fruit flies.
Absolutely. And when other researchers finally found physical, molecular proof their genes were moving, they had to go back and check McClintock's meticulous, decades-old cytogenetic observation.
And the validation was perfect.
It was perfect. ASCII and Ds were cloned, and they were definitively shown to be class II DNA transposons. This provided the molecular proof that Ds had a mutation in its transposis gene.
Meaning it requires the functional transposons produced by AC in order to move.
Exactly. That molecular reality matched her genetic observations from the 1940s flawlessly. Ds literally could not move without the enzyme from AX.
And this molecular confirmation forced the scientific establishment to recognize not just her technical brilliance, but her profound insight into evolution.
Yes, because McClintock had understood something critical that was later confirmed. Transposons typically do not activate and move randomly in a healthy, stable cell.
They move when the cell is under intense stress.
Intense stress, like X-ray radiation or when the BFB cycle causes rampant instability.
So the dynamic genome isn't just fluid, it's responsive.
It's responsive. When the organism is under an existential threat, the genome has a built-in mechanism to create massive, rapid genetic variation.
And while most of that variation is probably bad,
most of it is detrimental, yes, but some of it might be adaptive, giving the organism a chance to evolve its way out of the crisis. McClintock saw the genome not as a static blueprint, but as a reactive, self-editing system designed to promote survival.
once that validation was secured in the 1970s, the wave of honors began. Massive, well-deserved, if 30 years late.
Started rolling in. She received the National Medal of Science in 1970. She was the first woman ever to receive it.
And a MacArthur Foundation grant in 1981.
And then came the ultimate recognition. The Nobel Prize in Physiology or Medicine in 1983. She was 81 years old.
And the Nobel Foundation awarded it to her alone for discovering mobile genetic elements.
And the Swedish Academy of Sciences, in its citation, made this rare historical comparison. They likened her scientific career arc, discovering a profound principle that was rejected, only to be resurrected and celebrated to that of Gregor Mendel.
Wow. McClintock's complex journey, though, the early breakthroughs, the isolation, the ultimate triumph, it's naturally led to these competing biographical interpretations. It's created what some people call the McClintock myth.
Right, she became this powerful symbol of the genius outsider.
The initial influential biography, Evelyn Fox Keller's 1983 book, A Feeling for the Organism, it argued pretty strongly that McClintock's professional isolation, which was partly due to her sex, was actually integral to her success.
Keller's argument was that being an outsider allowed her this unique, holistic perspective that the mainstream, more rigid thinkers couldn't achieve. But that same status is what led to the hostility and rejection she faced.
And there are some striking historical anecdotes that certainly support that narrative of a brilliant, uncompromising outsider clashing with the mainstream.
Oh, yeah.
For instance, the highly influential geneticist Sewell Wright apparently suggested that she just didn't understand the underlying mathematics of her own work.
He viewed her visual, qualitative approach as somehow mathematically deficient.
And then there's the famous story involving Joshua Lederberg, a Nobel laureate himself.
Lederberg visited her lab at Cold Spring Harbor with some colleagues, and McClintock, who was known for her impatience with arrogance.
She reportedly threw them out after only half an hour.
She did. And Lederberg reportedly came away saying she was either crazy or a genius.
So why would a prominent figure like Lederberg, a respected scientist, say she was either crazy or a genius? What was so radical about what she showed them?
I mean, you have to think about it. She was showing them a level of complexity and dynamic activity in the genome that just contradicted the entire established concept of linear inheritance.
It was too chaotic.
It was too chaotic, too visually demanding, and it required a complete reordering of their intellectual framework. For someone steeped in the old dogma, her work had to be either delusion or pure brilliance. There was no in-between.
But this narrative of wholesale marginalization was later challenged.
It was. A revisionist view emerged in 2001 with Nathaniel C. Comfort's biography, The Tangled Field.
And Comfort called the rejection story the McClintock myth.
He did. He argued that while she was certainly unique and sometimes isolated, she was actually well regarded by her professional peers, even during the period of her transposition discovery. His argument is that the isolation might have been more of a self-imposed distance from the mainstream.
Driven by her famous capacity to be alone and her intolerance for intellectual arrogance.
Exactly. Regardless of how we frame the source of that isolation, was it institutional marginalization or self-imposed distance?
Mhm.
Her cultural impact is undeniable. Her life story is just a testament to conviction against consensus.
And she continues to inspire generations. I mean, she's been featured on U.S. Postal Service stamps, memorialized in buildings like Barbara McClintock Hall at Cornell in 2022, and she's inspired works of fiction and plays.
Her story has become a touchstone for scientific integrity.
It truly is a remarkable trajectory. You go from the masterful technique that defined cytogenetics in the 1930s to this revolutionary solitary discovery of the dynamic, responsive genome in the 40s and 50s.
And then finally, the ultimate global recognition and honor decades later.
And that leads us back to her own words from 1973, reflecting on that long delay in acceptance. One must await the right time for conceptual change.
Her discovery of mobile genetic elements showed us that the genome isn't a static repository of code. It's an intensely responsive, dynamic system, especially when it's placed under stress.
So what does this all mean for you listening to this? McClintock had to transcend the static genome paradigm to see the truth. So think about your own intellectual field, your profession, or an area of interest where you're seeking new knowledge. What intellectual framework or idea currently dominates your view? What's the equivalent of the static genome theory in your domain?
What's the thing that might be preventing you from seeing the next fundamental pattern?
Exactly. What established truth is so deeply ingrained that it actively resists complexity? What idea requires a period of stress, or maybe just a willingness to adopt the perspective of a true outsider to finally be revealed? That, I think, is the legacy of the jumping gene.